I'm trying to, as some of you know, in the work package 2, we are developing some live papers. So we aim at giving to the neuroscientific community some kind of data models and analysis pipelines. So they can really use that data, they can really adapt their pipelines to fit the data with that data, and also use the models we are generating and all this. So in order to do that, I'm going to focus my talk about I'm going to split my talk into different parts. The first one is scientific research. I'm going to explain one of the live papers we are currently developing. I'm not going to get into a lot of detail in the live paper, but just to give you a bit of flavor of what it is about. And then I'm going to show you how these scientific researches can be integrated with EBRAINS in the form of a live paper. Okay, so as we have seen during yesterday and especially today with the talk of Johan Storm from the microscale towards consciousness, and also from Francesco and Andrea Pigorini, that we go up to more high scales, like from mesoscales with wide field calcium imaging and up to the whole brain scale. When we try to characterize the brain states, it's highly important that we try to study the global but also local dynamics and properties of these cortical brain states. With that, we try to allow researchers to link experimental and theoretical research, so we can design simulations, in silico simulations, that you can infer data to that simulation to try to fit the real world with the simulated one. But you can also do reciprocal testing of new hypotheses, so you can do some experiment in the real world, and then you can test it in silico, or you can do the same in silico and then try to see if the same happens in an experimental setup. We can also enable more extensive modeling research. So as you all know, with in silico models, you can explore conditions that cannot be met in real world. And you can also perform extensive parameter search studies. And finally, and I think that's the most important one, we can optimize the analytical tools for the clinic. So one of the important topics here is to quantify the brain state. So not only characterize, but quantify. In this case, it would be like the disorders of consciousness or also anesthesia levels, but also improving the therapeutic interventions, like using TDCS or planning of surgeries, as it could be to precisely localize the epileptogenic focus of an epileptic patient. So to do that, and I was saying before, these are some examples of publications. I would roughly say that it's not even 1% of all the total publications on brain states. You cannot see it now, but if we get it more into detail, there are lots of different brain states that can be analyzed. We can have slow waves, we have unresponsive wakefulness, we have slow wave sleep, we have lots of different brain states, but not only that, we have also different methodologies that can be used and different types of data that you can register, like you can do it in vitro cortical slices, you can do it in the whole brain, you can do neural simulations, you can use high-density EEG or PET or imaging techniques, etc., etc. So it is important that we try to unify all the available knowledge and to put in common all this knowledge into a unified framework by using, let's say, a unified platform or a unified platform where every researcher can use, and they can use the data, and they can collaborate with other researchers, and that is what EBRAINS is trying to do. Okay, and that's what we are going to demonstrate with the use of live papers. So again, some other examples. I'm sure you've seen at least the rightmost part figures. So when you're doing experimental experiments, like in the real world, and as I was mentioning before, we have different multiscale and multi-species and multi-methodologies. So on the left-hand side, we have the as the microscale, we have the cortical slices. In this case, it's in vitro and in a ferret animal. In the middle part of the slide, we have slow oscillations in cortical surface, like in multi-electrode arrays, which are placed in the cortical surface of, in this case, in the mouse. And we can also investigate the levels of anesthesia that are placed there. But we can also have intracranial, more of up to the macro scale. We can have intracranial LFP recordings also in different anesthesia levels. For example, this is in a rat in vivo, where we place anesthesia to the animal, and then we wait until the animal wakes up, and we try to characterize which are the stages that go from fully unconscious to fully conscious. Okay. And not only that, not only experimental setups, but also in silico setups. So we can do exactly the same from the microscale towards the macroscale. So in the first image, we have a model which was developed by Leonardo Della Porta, and we try to replicate a cortical slice. We can try to, by modulating some of the parameters of the model, we can try to generate different brain states. And from that, the infinite range of possibilities you can do by varying these brain states, these parameters, and obtaining different brain states is infinite, as I was saying. So you can try to then apply different kinds of metrics that allow you to properly characterize in an in silico setup what are the different brain states. Then we have the wide field, like in the mesoscale, we have the wide field calcium imaging, where we can study not only the responsiveness, but also the spatial propagation of slow waves. And finally, as Professor Destex showed yesterday, we can have whole brain simulations, not only about human, but we can also have different species like mouse and macaque, where we can replicate synchronous and asynchronous and many other different brain states. So let me just get a little bit more into detail in the microscale. So for example, what we're trying to do with the live paper is try to merge both experimental and also in silico setups. So this is a clear example of a paper published last year from Barbero Castillo that we can get many different brain states in an experimental setup, and from the model from Dalla Porta, we can then replicate them in an in silico setup. So that is something that by having this slice in both regimes, in the synchronous one and the asynchronous one, we then are able to study the effect, for example, of enhancing or blocking different channels or receptors of the neurons. And with doing so, we can then study the properties of the network, such as waves propagation or PCI. If we then go to the in vivo preparations, and I was showing, as I was showing before, this is a paper also from 2020, I think, from Da Silva, where we use the surface multi-electrode arrays, which are surface electrodes, and we try to see we try to characterize what is the level of anesthesia in the rat, and we try to see the properties of these different levels of anesthesia and what are the differences shown in these different levels of anesthesia. And finally, up to the whole brain scale, this is in silico. By using these whole brain models with different species connectomes, we're also able to reproduce both synchronous and asynchronous, asynchronous like wake-like and synchronous like sleep-like or anesthesia. So by using these techniques, in this case it's DBV models, and as you can see, the left part of the video is asynchronous and the right part of the video is slow oscillations, as you can see. By doing so, we can then explore the whole different range of metrics that can be computed here, okay, like, for example, PCI. So that's the scientific part of the talk. Now I'm going to the integration, the technical integration in EBRAINS. So Shailesh, I don't know if it's here. Hello, Shailesh. And Andrew Davison have developed the EBRAINS live paper service. So EBRAINS is trying to offer to the neuroscientific community a way in which you can publish your papers not only in a static way, but also in a live way, where you can include all the information of the paper there, but you can also allow the user to use the data, use the models used in the paper, and also use the analysis pipelines. And this is a clear example of what you can do. And we're trying to extend this service by providing kind of an added workflow to this live paper service, which is going to the EBRAINS collaboratory and making use of the Jupyter notebooks, where you can access the data, you can access the models, and you can access the analysis pipelines. And with that, it's poorly shown, but as you can see, you can kind of develop a GUI where you, by varying some of the parameters, you can obtain different figures that are shown in the static paper. So the main question here is, what is the added value of live papers? So there are lots of them. Some of them are here. You can reproduce or modify the existing analysis pipelines. You can store your data and enable this data reuse. You can get new collaborations and new funding opportunities. You can increase the visibility of your work. You can also get a citable DOI and run your models on HPC. And finally, you can, instead of just reproducing, you can also integrate with the help of the EBRAINS technical coordination. You can also integrate your analysis pipelines in the platform. And that is the scientific talk that I showed before. Now it's being implemented in EBRAINS. So all the videos and figures that I've shown, these are real live figures that are going to be shown on EBRAINS for this live paper. So we can find the data and the models, we can rerun the analysis through EBRAINS, and we can also run simulations and get different results. For example, for wave propagation to the whole brain scale, or more to a mesoscale, we can also see how wave propagates through different levels of anesthesia. And that's all from my side.